Hi, everybody. First of all, I would like to thank the organizing committee for this kind invitation to present the work of my lab today with you at the second edition of the United Scientific Aging Conference in Munich. I would have loved to be with you in person today, but unfortunately, I had other obligation which forced me to stay in Montreal. But anyway, I'm very pleased to be here today by video conference to present you one of the latest work done in my lab by a very talented PhD student, Audrey Guittard, that I co-supervise with my colleague, Dr. Valérie Monret. So today I'm going to talk about the electrocorticographic and astrocytic signature of stearoyl-CoA desaturase inhibition in the triple transgenic mouse model of Alzheimer's disease. Like you probably know, Alzheimer's disease is the most common cause of dementia among people over 65 years old. And it represents 1/3 of the people over 85 years old. And at the moment, there's around 55 million people suffering from this illness. There's a huge socioeconomic burden associated with this illness. So if there's no cure found before 2040, there will be almost $300 billion per year that will be necessary to take care of these patients. And by 2050, we should have more than 100 million patients if no cure is found. At the moment, there's no treatment to cure or halt this neurodegenerative disorder. And this problem is only going to increase because of the aging of the population, particularly in industrialized countries. So one of the most common problems encountered during aging is memory loss. So at the preclinical stage, people can suffer from subjective forgetfulness complaints. And if it worries the person, there's a greater chance that these problems will turn into amnestic cognitive impairment, MCI, a prodromal stage of Alzheimer's disease. An individual with amnestic MCI commonly progress to Alzheimer's disease, converting from one diagnosis to the other at an annual conversion rate of 10 to 15% per year on average. So for many people, MCI is the earliest manifestation of AD, but not all of those MCI will convert to AD or another dementia. So this is why we need to better understand the cellular molecular alteration that leads to memory impairment during aging. Among the most prominent neuropathological markers of Alzheimer's disease, you have amyloid beta pathology, tau pathology, and neurodegeneration. Amyloid beta is derived from protein called APP that will and A beta will accumulate and form plaques extracellularly in Alzheimer's disease. You also have the tau protein, which can bind to tubulin to stabilize the microtubules. But in Alzheimer's disease, we know that tau gets hyperphosphorylated and will tend to form aggregate called neurofibrillary tangles. You also have neurodegeneration, and at the onset of the disease, there's some brain region that are affected particularly, like the entorhinal cortex or the CA1, both regions that are important in memory formation. And by the end of the disease, neurodegeneration is visible globally throughout the brain. On top of those neuropathological markers, we also know that in the early stages of Alzheimer's disease, people are also suffering of sleep perturbation, such as difficulty initiation sleep, poor sleep quality, decreased sleep duration, insomnia, excessive daytime sleepiness, and also circadian alteration. And we know that all these factors can influence and participate in memory decline at the onset of Alzheimer's disease, and this will progress during the course of the disease. Thus, to study the molecular and cellular mechanisms underpinning sleep perturbation, we decided to use the triple transgenic mouse model developed by the group of French astronomers at the University of California. This is one of the most used and characterized animal model in the field of Alzheimer's disease. But surprisingly, a detailed analysis of sleep features has never been performed in this well-studied 3xTG mice. In our study, we use female 3xTG mice because they show worse phenotype than male regarding A-beta pathology, which is in line with the higher risk of developing Alzheimer's disease in women. We also used 3xTG mice between 4 and 5 months old because we wanted to investigate sleep perturbation at the onset of Alzheimer's disease when cognitive impairment starts to manifest and correlate with the accumulation of intraneuronal A-beta and the hippocampus. We also wanted to see if treatment could reverse sleep perturbation that we could potentially be observed in 3xTG mice. So while we were designing our experiment, there was a nice paper published by our colleague, Dr. Cal Fernandez, that just came out in Nature Neurocommunication, showing that the inhibition of stearoyl-CoA desaturase, or if you prefer SCD, which is a lipid modulating pharmacological treatment, was able to reverse cognitive, synaptic, and immune deficit in the 3xTG mouse model. Thus, we wanted to see if this treatment could also reverse sleep disturbance in these mice. What some people may not know is that abnormal accumulation of lipids was described for the very first time in the first report on Alzheimer's disease by Alois Alzheimer itself in 1906. And in the recent decades, a variety of Alzheimer's risk factors have been found to be involved in lipid metabolism. Recent findings suggest the involvement of dysfunction in lipid metabolism, such as oleic acid buildup in the brain of AD patients and also in animal models. Specific phospholipids were found to be increased in different brain regions and decreased in the plasma of Alzheimer's disease patients. And an elevated level of monounsaturated fatty acid, MUFAS, was reported in AD brain. Interestingly, this elevated level of MUFAS was associated with higher mRNA expression of SCD, the rate-limiting enzyme in the biosynthesis of MUFAS. Thus, with my colleague, Dr. Valérie Mongrain, and our PhD student, Valérie Audrey-Card, we decided to look at the electrocorticographic, or if you prefer, ECOG, and astrocytic signature of SCD inhibition in 3xTG mice. So 4 to 5 months old 3xTG mice were implanted with the osmotic pump for SCD inhibitor treatment into the ventricles, and with electrode for electrocorticographic and electromyography analysis. Following 28 days of treatment, ECOG and EMG signals were recorded for 24 hours, starting at the light onset. In the bottom left of the slide, you have an example of the ECOG and EMG signal that were recorded during awakening, but also during paradoxical sleep, or if you prefer REM sleep, as well as slow wave sleep or non-REM sleep. So why studying astrocyte in more details? Well, first reason is that in the brain, astrocyte regulates the synthesis, distribution, and storage of specific lipids. Secondly, alteration in astrocyte and their function were reported in AD patient and in animal model. Finally, we know that astrocytes have been implicated in the regulation of sleep, more precisely in the regulation of EKG activity during different sleep states. What is also interesting is that higher expression of activated astrocyte marker genes in the prefrontal cortex was associated with sleep disturbance and cognitive decline in humans. However, although we know all this, the relationship between sleep disturbance, astrocyte, and lipid metabolism remains to be explored in AD. What we first observed is that the wake-sleep architecture is altered in 3xTG, shown here with the red bars, when compared to wild-type animals, the blue bars. Indeed, 3xTG mice were found to spend significantly less time awake and more time in slow-wave sleep during the dark period. These effects were not significantly impacted by the inhibition of SCD. Interestingly, 3xTG mice show more individual boots in the wake and slow wave sleep states, which mean higher fragmentation, or if you prefer, less consolidation of vigilant states, which is an indicator of poorer quality of sleep. The bipolar and unipolar EEG signals were submitted to standard spectral analysis using fast Fourier transform to compute spectral power between 0.5 and 50 Hz for each state. The spectral profile of the EEG within the different regional states interrogate using a standard spectral analysis can be considered as indicator of sleep quality. Standard bipolar absolute activity informs on change in the organization of the cerebral cortex. Here we observe a significant decrease in spectral power for a wide range of frequencies for the treat vigilant state in the 3xTG mice in comparison to wild-type mice, which indicate a global reduction in synchronized cortical oscillation in 3xTG mice in all vigilant states. Standard bipolar relative activity informs on the relative contribution of the different frequency component of the signal. 3xTG mice present major alteration in the relative contribution of different frequency bands during all three vigilant states, alteration that are not significantly rescued by the inhibition of SCD. We then aim at verifying whether the inhibition of SCD in the 3xTG model affect EEG activity in a brain region-specific manner. So here you have the black dots on the top of the graph indicate the 0.25 Hz bins with significant difference between genotype. Red dots indicate bins with significant genotype by treatment interaction, and the gray dots indicate bins with significant difference between treatment. So if we start with the motor cortex, you can see that activity was importantly affected by genotype in all states during the light and dark period. In general, mutant mice showing less activity below 20 Hz and higher activity above 20 Hz in comparison to wild-type mice here in blue, except for the paradoxical sleep, for which activity between 7 and 10 Hz was significantly higher in the mutant mice. In the visual cortex, during wakefulness, 3xTG mice generally express higher activity in lower and higher frequency, but less between 7 and 10 Hz in comparison to wild-type mice. During slow-wave sleep, 3xTG mice show significantly less activity than wild-type mice below 20 Hz, but more above 26 Hz. In paradoxical sleep, 3xTG mice also had a general more activity than wild-type mice in multiple Hz bins between 0.5 and 50 Hz. So these findings indicate that 3xTG mice show a highly specific spectral signature that also depends on frequency, state, and brain area, and that the SCD altered synchronized activity in a genotype, frequency, state, and topographic state in dependent manner. Since there's gliiosis in the brain of 80 patient and in the 3xTG mouse model, we next ask if inhibition of SCD could reverse astrocytosis in the hippocampus, the thalamus, and the lateral hypothalamus. We observe an elevated number of GFAP stem cell in the CA1 and CA3 region of the hippocampus in 3xDG mice that can be rescued to wild type values in the CA1 by the inhibition of SCD. We also found a marked influence of genotype on ALDH1L1 staining in the lateral hypothalamus as well as in the thalamus, two brain regions important for sleep regulation. But these effects were not modulated by 28 days of SCD inhibition. This figure showed that SCD inhibition significantly increased the radius size of the area covered by astrocytes and a tendency to increase the mean length of astrocytic process and branches. Thus, these observations suggest that the inhibition of SCD for 28 days modestly increased the length of individual astrocytic process, resulting in a generally larger diameter of coverage. So, in summary, you observe a reduction in time spent awake and increase in time spent asleep in slow wave sleep during the dark period in the 3xTG mice, but these effects were not restored by inhibition of SCD. We also observed that ECOG activity was markedly altered in triple transgenic mice for all wake and sleep states, and there was alteration by inhibition of SCD in different manner in the triple transgenic mice model versus the wild-type mice. Finally, we observed that astrocyte density is higher in the hippocampus as well as in the lateral hypothalamus and in the thalamus in the 3X mice. And this effect was rescued by inhibition of SCD in the CA1. To finish, I would like to thanks all the members of my lab, especially Audrey Hector, who lead this project, and also Dr. Valérie Mongrain, Carl Fernandez, and Jean-Marc Lina, and the granting agency for supporting this work. So thank you very much for your attention, and I'm here to answer your question. Thank you very much, and have a good day.